sense. Want everybody to know that before.
I'm from, well, my parents are people.
All right, let's talk a little bit as a class, just summarize this. We can kind of go down the row. So we'll start over here by this door. So what was this paper about? Like, what were they asking? I think most of these questions can be answered by the abstract. So, yeah, so they were looking for the ancestral flowers. Anybody catch why they wanted to do that? Why they would have to do that?
Basically, they just said that there wasn't a lot of information about stuff there, like further back.
Yeah. Anybody who read that section about like the timing, right? It looks like there was about like 100 million years essentially where they were flowering plants and had no flower off. They were kind of curious about where this started off to understand a little bit about how Angiosperms evolve. And evolution often pairs along with like development. So in modern organisms, the way things develop often sometimes sort of recapitulates their evolutionary history. So that can be helpful for living things as well. All right, so down the list, what kind of data were they using there?
Oral traits.
Yeah. So how do you think they got that?
They used a bunch of different data sources, personal observation. And so there were a few personal observations.
Yeah, it looks like there's a database too, maybe.
Something like that.
Yeah, some giant spreadsheet. Hopefully not a spreadsheet for the number, but possible. All right. Next group. Does this flower still exist? That's well. Probably not, right? Yeah, we were just picking between flowers we found outside. All right. What was uncertain about this reconstruction? Yeah, so they were looking for fossils and better information on divergent states to improve their analyses. If you guys saw the picture of their imaginary flower, not imaginary, their theoretical flower, some of the traits that they put in this picture, like they had less confidence on. Did anybody catch which those were? Node depth. Yeah. And also like the number of organs and like the number of like whorls they have. They got a lot more than for a lot of flowers. And it looks like this is bigger than about these questions. Anybody want to tell me whether it was actinomorphic or zygomorphic? Yeah, that's mainly to practice those vocab terms, right? What about, was it bisexual or unisexual? Yeah, bisexual. Okay, cool. So some of these traits we're going to see in at least one of the groups that we're looking at today. Okay. So we're going to start out with our extant, so existing, alive today, most basal angiosperms. So we're talking about things that have the earliest divergent state first. So it's called the Anagrade or sometimes the Anagrade group, just an acronym. So we're going to have Lorealis, Nymphacia, and Austrobailealis. Are three big orders here that we're kind of lumping together. And there are two other sort of smaller groups for the I and T, which we'll see on the next figure. So thinking about the word grade, just an explanation here. So little systematics torture again. So looking at this figure, so I've circled out our anagrade families here. So the Lorealis, the Nymphacia, and these other groups. So we have terms that we can apply to phylogenies. So on the right, I've pulled up a reminder of what it might mean that something is monophyletic, paraphyletic, or polyphyletic. Anybody want to tell me which of those best matches how we have those anagrade families splitting off? Close. So if you look here on the image, so a monophyletic group is going to be any group that comes from a common ancestor and includes everything descending from that common ancestor. But what do you see here on this figure? So if we have a split here, say this is our common ancestor, we're looking at this group, does that include everything? No, right? We're keeping out a lot of the descendants of that last common ancestor. So this is actually an example of paraphyly. Polyphyly would be if you picked like random groups here. This paraphyly is where we've just kind of excised, cut out a big branch here. Yeah. The reason I'm bringing this up is that's why it's called the anagrade. So grade is kind of like a taxonomic term. So it's telling you that it's not like one monophyletic group, but they are all kind of related to each other. So that's where that term comes from. Okay. So we're going to just walk through some of these species. So the first one we're going to talk about is something called Ambrella. And this is actually only one species in this order. So it's this first one branching off. It's a really long lineage to get to just one remaining species. And it's found practically nowhere. It's in one island in New Caledonia, so in the South Pacific. It grows kind of in the shade, it's a little shrub. So this is our earliest split. This is going to be our sister group to all our other angiosperms. And it's kind of weird. So it does have wood, but its vasculature is kind of different than our other angiosperms. We saw that that was one of the differences between gymnosperms and angiosperms. You remember way back to when we briefly looked at that in our gymnosperms. And another thing that's kind of cool about Ambrella is that it had a lot of horizontal gene transfer, as far as we can tell, with algae, with lichens, with mosses, kind of all over the place. So if we look at the flowers, someone has described them as nondescript and not very pretty, but I actually think those are cool looking because they're so weird, but they're small. They're really small, they're in clusters. These form these little clusters of drupes here. Okay. And about a decade ago, they actually awarded an NSF grant for $11 million to sequence the genome of this. So looking at where Ambrella sits relative to our other angiosperms, why do you think they might have done that? Like, why would they bother? Yeah, potentially to figure out if it's supposed to be grouped with something else, or maybe if it's not an angiosperm. But if you'll also note, so look here, how would you describe that relationship between Ambrella and all these other things, the rest of our angiosperms? Yeah, that branching point is more basal. And this is actually a sister group to everything else, right? So at this split, we can see Amberella going off on its own, and then everything else is coming from a different common ancestor here. So this can act as a really good outgroup if you're looking at the rest of the angiosperms. It's a good point of comparison, right? Something else, something weird. So if you're trying to make estimates about what happened in the angiosperm timeline, knowing that we have this weird separate branch can be really helpful. A little bit more about flower evolution, right? Most of our plants now are flowering plants, or at least are very economically important ones. We've had this explosion of angiosperm diversity. Yeah. But it's kind of confusing, potentially, to figure out where they came from, right? Did they just come out of nowhere? So people have looked into those genomes to try and figure out what happened. And an interesting thing that they've noticed is that plants regularly show evidence that their whole genome has been duplicated. So instead of going from diploid to haploid and then having two haploid cells come together to form a new diploid one, they just got two diploid genomes, smashed them together, and ended up with like 4 copies, essentially. So one thing that they think has happened is that there was one of these events right before our angiosperms diversified. So they got a whole extra set of all their genes, meaning that there's kind of less selection pressure potentially on these species then, because they have their original set essentially to do all the functions of the plant. So they have all these extra genes hanging around that they could kind of like play with. So that's one of the theories as to why we were able to have so much room essentially for flowers to diversify and change, and these groups to become so different from each other. There are plenty of other whole genome duplication events in plant history, and actually we do try to duplicate through cultivation parts of the plant genome as well. So polyploidy, so increasing the ploidy, diploid, haploid, there's actually more, is something we do with crops. I think I read somewhere that that's like how they made strawberries bigger, right? The wild ones are actually really tiny. So I have to find that source if I'm going to say that officially, but I think that's one of the ways they did that. Okay. So going back to Amberella, in that article we just read, we had a bisexual sort of whorled flower. But Amberella, this early branching point, doesn't actually look like that, which is fine. It's had a lot of time to evolve. It's the only remaining species, but that doesn't mean it necessarily is fully unchanged. So Amberella does have unisexual flowers. So it has both female flowers and male flowers that look pretty strange. So you can see here these weird sort of things that almost look like petals where they have their anthers. So these are actually the stamens of the flower. So instead of having that filament, that little stalk and the anther on the end for pollen, we've got something like this. We also have sepals here. So this is a word you'll see when something doesn't have sepals. If you notice the phrase like undifferentiated perianth in that article, they're talking about the fact that we don't have green sepals and then colored petals. We've got kind of like one structure here. And in the female flower in Amberella, we can actually see that it has almost what would be a stamen. It's the sterile and non-functional, which is kind of weird. My understanding is that Amberella can also change sex at certain points, which is cool. It's a weird plant, even though we can't find it. So more notes about genetics of Amberella. This is weird. So mentioned on that previous slide that it's experienced horizontal gene transfer from other things in the same environment. So I just wanted to kind of point out what that is and mention a couple of ways that that could happen. So horizontal gene transfer is when an organism is getting genetic information from something else, right? So not just inheriting genes from its parents, that would be vertical gene transfer, like up a generation, down a generation. So horizontal gene transfer is from something else in the environment. And this definitely happens in biology. It happens a lot in prokaryotes. And this is actually how we think that eukaryotic cells ended up with like mitochondria. We also have plastids. And in plants, we have our chloroplast, right? The idea is that we had an original cell that like ate another cell and then developed a symbiosis instead of digesting it, essentially. Yeah. So it's less common in eukaryotes, which are more complex, but we do see it, and we see it a ton in Ambarella. So here on the right side, I pulled out from an article some of the genes that Ambarella got from other places here. So exactly how this happens in plants is not necessarily always clear. So the most logical sort of part, at least to me, is the idea that it comes from parasites. There are parasitic plants that infect each other, or viruses, bacteria infect plants just like they do animals. Fungi are also parasitic. So one of the sources of foreign DNA can be from those parasites, but instead of getting destroyed or destroying the plant, they actually get a little bit incorporated. In cultivation, so like when we're growing things on purpose, this can also happen when we graft things. That's when you like cut off a piece of a branch and stick it onto another plant, and they kind of merge together. You can end up with really weird trees that grow all sorts of strange fruits by doing this, or apple trees that grow multiple different kinds of apples, things like that. Other ideas are sort of part of the food chain, so cells consuming other cells. Also, plants have a number of transposable elements in their genetics, and this is a potential source of foreign DNA, although exactly how that would get incorporated, I'm not totally sure. Why Ambarella has so many, even compared to other plants, is not super clear. A quick note about transposable elements. I am not a geneticist, but I found a picture that I could somewhat understand. So if you all know a lot more about transposable elements than me, please tell me. But transposable elements, little sections of the genome that can move around, essentially. So they were often a large portion of what we thought of as junk DNA, but they can get like cut up and inserted places, and they can change the function of genes. Sometimes they're triggered by stress conditions. So in this figure, we can see that there's a stress response to, I guess, this lightning strike here. Maybe that's their stress response that they're showing. So under unstressed conditions, the plant is healthy and fine. Under stress conditions, it might wither and start dying. But if this stressful event triggers our transposable elements move, it gets inserted into the genome, moved around. It can change how the genes function and create a plant that remains healthy under stressful conditions. Yeah. So the fact that these things are very present throughout plants, and that plants undergo stress, is one idea of how we might end up with this horizontal gene transfer in plants. And this is my question to you, since I'm sure some of you have taken genetics. Did you talk about this in genetics class? And do you know other facts about transposable elements that I should know that might be relevant here? No? Okay. I'll go ask Dr. Temke if she knows stuff that you guys should know, but that's what I got for you. All right. So weird genetic things going on in Ambarella, which is kind of cool, in addition to the fact that it's a sister group to all of our other Angiosperms and makes this nice outgroup on the side. Okay. So we're going to walk our way in to the rest of the Anagrade. So next we're going to look at the Nymphaceae, or the order Nymphalis. So Nymphalis is an order, Nymphaceae would be a family. Okay. So Nymphalis, this order, actually has 3 families within it, mainly aquatic plants. The one that you'll probably encounter or notice most frequently are the water lilies. So they are cultivated because they're really pretty. And some of them are like actually really crazy. This is a real photo. This isn't Photoshop. So some of these Water lilies can grow up to 2 meters in diameter. So there's apparently some park where you can go sit on them if you want. They have incredibly strong vasculature. So they've actually used this to kind of come up with better engineering designs to look at this branching pattern of the vasculature and some of its properties, because these water lilies, when they get rained on in the Amazon, are carrying a bunch of water kind of on top of them, and they have to withstand that, as well as current and other things. So when you're thinking about a water lily, it actually is attached to the pond or the stream bottom, so it is kind of anchored down, but the leaves and the flowers are just kind of floating on top there. It's got good light from the surface of the water, probably, and nutrients coming down from the roots, I guess up from the roots. The other group, or the other order in our anagrade, are the Lorealis. So the one that you might encounter in your real life is Anise. So this is a star anise seed pod cut open. It's pretty. I really like anise, but I think it might be kind of an acquired taste, kind of licorice flavored. There are about 100 species in this order. They're native to Australia, Southeast Asia, Pacific in general. So the star anise, its genus species name is Illicium verum. Used for flavoring, perfumes, and herbal medicine. Species in this order are 7. In general, they're going to be woody trees, so trees, shrubs, so shorter like bushes branching, and lianas, which are basically vines, essentially. They have simple leaves, often they're aromatic, and they don't have stipules, which were those little extra sort of leaves at the base of the leaf where it attaches to the twig. When you look at their floral organs, so their androecium, their gynoecium, they're going to be spiraled. So they look a little bit like that first flower we looked at, but not totally, right? That was in a whorl rather than a spiral. Okay. So that's our anagrade, pretty simple. The other thing we're going to talk about today and take a look at are the magnolias. Okay. So here we have a chart of those. The reason we're doing that is because they're kind of inserted between our 2 main groups of plants. Most of the plants that you will see out in the world and talk about are either monocots or dicots. Magnolias are kind of branching in between. Okay. So looking at this chart, anybody want to hazard a guess as to what our magnolias are going to look more like? Yeah, so their genetics might be more similar to the monocots. That's why we estimate the divergence like this. If we look at the venation specifically, though, we'll notice that if you were just like out in the forest, you might think that it was a dicot because of some of these more visible patterns. So the monocots developed parallel venation along their lineage here, but the magnolias split off separately before that happened. So back in the day, before they did all these phylogenetic studies, you might have grouped this in with the dicots traditionally. And that's part of why we have to call the dicots eudicots or eudicots, true dicots, because this group, to make it a monophyletic group, we have to kind of exclude the magnolias. So this is one of the places where our phylogenetic reconstructions have been changing. But here we have some pictures of some lovely magnolias, including actual magnolia trees, conveniently. So when you think magnolia, think actual magnolia. So they're going to be both woody species, so wood, die back, come back again next year, and herbaceous species, so where we have just kind of like green tissue that'll die back. And we have 4 orders, but 18 families, so we're just going to briefly look at these 4 orders for this section. Okay. So they used to be considered dicots. Sometimes they're going to have 2 cotyledons. Some of them will have up to 4. So there's a little variation there. And our modern phylogenetic techniques show that they actually branched off potentially more closely to them. So kicked them out of dicots and had to come up with a whole new word. Kind of a pain. Now, if you look at a magnolia flower as an example, you might notice that it looks a fair bit like that figure we saw in that first flower paper. And it is true that when we look at magnolias, a lot of their features are going to look kind of primitive. They're going to look like what we think the last common ancestor of the Angiosperms look like. So when we look at the inside of the flower, we're going to see that the carpels are not fused together. We're going to see that they either have a ton of petals or they don't really have any petals. There are also differences between different plants on what their pollen grains look like and what pollen sacs look like. We haven't talked about that really, so I'm going to leave out those characteristics for our magnolias because I also don't think that you're likely to go inspect those out in the field while you're attempting to do plant collections in the snow. But if you get super into botany, pollen's important. Magnolias are more common in tropical climates, but there are species found pretty much everywhere. So our first order right here is our Magnolialis. So this is traditionally considered the most primitive. So you can see that this flower really does look an awful lot like the flower in that paper. So when you're trying to identify things, you see something that looks a lot like it has these primitive traits, this is a good group to consider that you might be seeing. So it's going to have large leaves with the venation pinnate. So pinnate looking kind of like a feather. So we can see that here. Looking at this flower, does this look unisexual or bisexual? Some flower practice for us. Yeah, 50-50 shot. Anybody want to volunteer? So it's bisexual. So that's all right. It's all right. So the way you can tell, right, when we're talking about unisexual and bisexual, this is helpful, right? So we can figure out what they're talking about. So we're talking about like, does it have just male parts and just female parts? Or do we have both in one flower? So this is like that perfect flower, imperfect flower stuff. So what we can see here is that we have both carpels. So those carpels where we have an ovary, we have the stigma, those structures. And we also have stamens, which was where we had the male parts, the anther, the pollen. So because we have both of these parts here, we consider this flower bisexual because it kind of self-pollinate even potentially. So that's what we see here in this cone. Two things kind of clustered together in the center of the flower, as well as a bunch of petals around it. We actually do have some sepals apparently on this particular one. So some things you might encounter in real life potentially. Actual magnolias. Nutmeg is also in this group. So it's part of the order Magnolialis. Cherry moya, never eaten this, but I would like to. Looks exciting. Pawpaws. Based on the Jungle Book, I thought these were only faraway places, but apparently they are also native to the US, or the southern US. So I would also like to go try those. We'll see if I do a road trip this summer. Has anybody eaten a pawpaw or a cherry moya? I'm just really curious what they taste like. Anybody know? Well, if I find one in the next couple weeks, I'll bring it in and we can taste, but it doesn't feel too likely. Okay. So the sister group to the Magnolialis is going to be the Lorealis, actually named for laurel flowers. And some places describe this as a group of aromatic oil flowers, which sounds both kind of cool and potentially kind of gross, honestly. It does have a lot of uses. So plants in this order are economically kind of important. We have medicinal plants here. We have plants that are good for lumber, essential oils, and make good ornamentals as well. So some common examples that you might have in your kitchen would be cinnamon is in this order. Avocados are as well. And a more medicinal one is like camphor. I pulled out camphor here. That's kind of the smell in like Vicks Vapor Rub. I pulled out the molecular structure for cinnamon. So these are really abundant trees in moist, warm tropical areas. They're generally woody. So cinnamon is actually the bark of this woody tree. And they have cool chemicals in them. So they have secondary chemistry, and specifically the Lorealis have these aromatic oil cells inside. These aren't characteristics I picked out here on the slide, but you can also see that those leaves look a fair bit like our Magnolia leaves did, right? We have simple leaves, look like they're pinnate, just kind of boring, plain looking leaves like you might draw if you were, in third grade trying to draw. More examples. So laurel leaves is where they get their name. So laurel wreaths awarded to the ancient Olympians. Sassafras leaves are weird. They're shaped kind of randomly, even on the same tree. They have different numbers of lobes. That's also in this group. I picked it out because I really like sassafras. I'm from New Jersey, and it grows everywhere in every field that has ever been mown. It's like one of the first plants to come back. It tastes funny, which is exciting to me. I like things that taste weird. And also, it's banned like in the US. So sassafras trees used to be used commonly to make root beer. My mom always told me that I could dig up the one in the backyard, use its roots, and make homemade root beer if I wanted. I didn't want to kill the tree, so I didn't. But they don't anymore because they banned saffrol, the oil in this plant, in 1960. This also caused a huge stir because gumbo, like in Louisiana, like one of the main ingredients to make it gel up is the dried leaves of sassafras. So they have an exception now for their dried leaves. I think the reason they gave for banning it is it caused cancer in some rats, although honestly, the hype about that is pretty high compared to plenty of other legal things. High doses of saffrol, though, are hallucinogenic and used in the manufacture of MDMA. So I may be starting rumors, but I feel like that would be more of a reason the government might want to ban it. So there's a structure of saffrol and a picture of a little sassafras plant and some tea someone was making. I don't think it grows around. I was surprised about. Okay, our next group, Piperalis. So this comes from pepper. So black pepper, Piperalis pepper. So these are also going to be aromatic plants. They're going to be fragrant. They're going to be spicy usually. They have really weird flowers. This is really characteristic. So they have these inflorescences that just look kind of like spikes, right? You wouldn't even really guess that this was a flower if you saw one in person. I bit one in the jungle once. I don't know what the plant was. It was very peppery. I'm maybe allergic to pepper. It was a bad idea. We'll see if I can find any in any garden stores around. I'll do some Googling before I let you try biting them as well. But my tongue stung for like 2 days. But it was really fun. So Piperalis, we'll take a look at some pepper in the future. Okay. Finally, the last order in here is the order Canallalis. They're not very important. They're found mostly in the southern hemisphere, but their leaves would also often have a peppery sort of taste, which we can imagine why that might be the case, given the properties of some of these other plants. I've also heard, separate fun fact, magnolias, right? We didn't talk about aromatics there, but I've heard they taste like ginger. So if they ever bloom here, I'm going to try to make some magnolia jam and we'll report back. Okay. So our final note is just some ways you could guess that you're looking at one of these plants, say on your practical. You're going to look for simple entire leaves, right? So nothing going along that margin, that border. The flower is going to have a bunch of pistils. They're not going to be fused to each other. They might be in kind of like a cone. Their stamens are going to be kind of flat looking. They may or may not have distinct petals and sepals, probably not. They're going to be in kind of a spiral. They're not going to be obviously countable. You may have a smelly kind of plant with essential oils. And if you were out in the wild, you would expect them to be pollinated by insects, not like mammals or birds, also not. So that's what we got for today. Okay, question for the class. Does anybody have an avocado tree at home? No. Okay. Just asking for Thursday. My avocado pits are sprouting, but they don't have leaves yet. So they're super easy.